The genotype of a locus refers to the allele on a chromosome in haploid cells or the alleles of homologous chromosomes in diploid cells. In diploid organisms, genotypes may be homozygous or heterozygous. Specific genotypes influence specific phenotypes, and so genotypes can be used to predict the phenotype. For example, a person may want to know if they could have the mutation causing late-onset Huntington's disorder while still being symptom-free to make informed life decisions, as this disease causes suffering and has as yet no cure. Information whether someone might have the allele causing Huntington's disorder could be obtained from pedigree information on phenotype. Pedigree data on phenotype has shown the mode of inheritance of Huntington's disorder to be autosomal dominant. In an autosomal dominant disorder like Huntington's, a person with the disorder, if married to a person who does not have the disorder or a family history of the disorder, would have about 50% of their children, either male or female, suffering from the disease. Unaffected parents do not transmit the traits, and the trait does not skip generations. So this young man stands a 50% chance of having Huntington's disorder. However, a more definitive diagnosis is now possible as studies on DNA from individuals in families with Huntington have linked specific mutations to the expression of Huntington's. Likewise, other inherited phenotypes may, through studies on large numbers of individuals, be linked to a genetic marker, and genotyping used in predicting the possibility of an individual expressing the phenotype. So genotypes may be inferred from both pedigree data on phenotypes as well as DNA-based methods. Genotyping based on DNA sequence uses technologies such as PCR and arrays or chips. The DNA technology used is partly based on the type of sequence variation, whether the locus retains its length, as in substitution mutations, or whether the locus changes in length, as caused by duplications and indels, that is, insertions and deletions. Of course, any DNA-based method first involves extracting and purifying DNA from the sample cells. Provided sufficient DNA of good quality is obtained, we can proceed to genotyping. Among the genotyping methods, polymerase chain reaction using 2 primers followed by electrophoresis may be used to genotype alleles of different lengths. PCR amplifies the polymorphic locus using primers complementary to the sequence of the flanking regions, which are generally the same in different individuals, and the sequence known from the reference human genome sequences. Electrophoresis then separates the amplicons based on size. Alleles present in the individual can be determined by comparing the DNA bands obtained from an individual to a simultaneously run DNA ladder. As seen, individual A has 2 bands and is a heterozygote with alleles 1 and 3. Individuals B and D also have 2 bands, but the bands are a different distance from the wells than individual A's bands. So individuals B and D, though also heterozygous, have different alleles, that is, alleles 2 and 4. In contrast to these heterozygote individuals is the homozygous individual C with only 1 band, allele 1. In another similar genotyping method, though PCR is still used to amplify the target region, one of the primers is fluorescently labeled, and instead of slab gel electrophoresis, capillary electrophoresis separates the fragments, and the alleles, instead of being observed as bands on a gel, are seen as peaks in an electropherogram. Locus 1 shows 2 peaks and is heterozygous, having alleles 7 and 8, while locus 2 shows 1 peak, being homozygous and having 2 copies of allele 10. While PCR with a single pair of primers followed by electrophoresis can identify mutations that cause size changes, some mutations do not cause size changes, so genotyping has to use a different method. One such method which is used for genotyping SNPs is T-ARMS PCR or tetra-primer amplification refractory mutation system PCR. This genotyping method involves amplifying 3 fragments of different lengths by 4 primers. A common outer primer pair amplifies the entire target from both alleles. Each of the 2 inner primers and an external primer produces an amplicon of a specific size as the outer primers are placed at different distances from the SNP. Each inner and outer primer pair specifically amplifies different alleles as their 3' ends are complementary to the SNP. An additional mismatch in the primer destabilizes the inner primer base pairing to its non-targeted allele. After amplification, gel electrophoresis distinguishes the different genotypes. Homozygotes show 2 bands, the common control fragment spanning the length between the 2 outer primers, and an allele-specific fragment, while the heterozygote shows 3 bands, the common control and both allele-specific fragments of different sizes. Sometimes a more high-throughput method is needed. Thus, the SNP chip or array technology may be used. This technology can simultaneously test many individuals' SNPs at specific locations across the genome. A SNP bead chip has multiple surface beads, each coated with many copies of a probe. The probe nucleotide sequence on each bead targets a specific SNP and ends one base before the SNP. To genotype an individual's SNPs, the chip is incubated with the individual's genomic DNA that has been amplified and fragmented. Fragments denatured, if complementary to a probe, anneal to it. Fragments not attached are washed off. The chip is next incubated with labeled dideoxynucleotides and DNA polymerase, so a single dideoxynucleotide complementary to the hybridized fragment at the SNP location elongates the probe. Then the DNA template and excess nucleotides are washed off and labeled probes are stained. If fluorescent tags on A and T are red while G and C are green, a laser-scanned image of the homozygote AA or TT is red, CC or GG is green, and a heterozygote is yellowish. Genotyping could also use PCR followed by sequencing, which would detect any kind of mutation within a region between the forward and reverse primers. To summarize, 1. Genotyping is the process of determining an individual's genetic makeup or genotype. 2. The genotype determining a particular phenotype may be obtained by studying the inheritance patterns of the phenotype in families expressing the phenotype, or studying the DNA polymorphisms associated with people expressing the phenotype. 3. There are various DNA-based methods of genotyping, including those involving technologies such as PCR, chip technology, or sequencing. Now let's check our understanding of genotyping with a few questions. You may pause the video while you think about your answer. Question 1. The genotype of an organism, A, describes its appearance, B, is written in words, C, is determined by its phenotype, D, is the combination of its alleles. The answer to this question is D, which is the combination of alleles at a locus. Question 2. Does this example of a study's results suggest a marker that can be used to predict if an individual suffers from genetic disease A? Answer. Based on these limited results, a marker is associated with disease A. Most people with disease A, as compared to those without, have a particular DNA band. this DNA band or marker is not randomly distributed in the population. Question 3. What about these results? Is there a marker to predict if an individual will suffer from genetic disease B? Answer. Based on these results, none of the DNA bands are significantly more often present or absent in people with disease B than those without. So further studies must be carried out to associate genotypes with disease B occurrence in individuals. Question 4. The diagram shows the inheritance of ABO blood groups. The blood groups of some of the individuals are given. What could be Ariana's genotype? A. AO, B. BB, C. BO, D. OO. The answer is C, a heterozygote B blood type, that is BO. As Ariana's granddaughters are blood types O and A, and since their father is blood type O, Ariana's daughter must have the alleles A and O. Also, since Ariana's son has blood type B and her husband has blood type A, Ariana must have blood type B. Ariana's daughter's A allele must thus be inherited from her father, so the O allele must be from Ariana, showing that Ariana's genotype is BO. Question 5. Based on the position of the SNP shown on the sequence and the bands shown on the gel, individual 1's genotype is A. Homozygous for A, B. Homozygous for G, C. Heterozygous, D. Unable to be confirmed. The answer is B. Homozygous for G. Individual 1 has 2 bands, so must be a homozygote. The band common to all genotypes must be the control that extends between the outer primers. The second band in individual 1 has moved the furthest, so it must be the smallest fragment. Allele G, amplified by primers 1 and 4, is shorter than allele A, amplified by primers 2 and 3. So individual 1 must be homozygous for G. Question 6. The electrophoregram shows the same 4 markers on the DNA profile for the mother, father, and child. At the D7S820 marker, the mother has only 1 peak labeled 10, but the father has 2 peaks labeled 8 and 10. Explain the significance of this observation. Answer. The mother is a homozygote at the D7S820 locus, having 2 copies of allele 10. The father is a heterozygote at the same locus, having alleles 8 and 10. Question 7. A pedigree and microsatellite analysis of a family with a history of early Alzheimer's is shown below. Which polymorphism is early Alzheimer's associated with in this family? Does the male, indicated by Z, carry the allele for early Alzheimer's? A. 1 yes, B. 2 yes, C. 2 no, D. 4 no, E. 1 can't tell. Answer. The allele present in Alzheimer's patients, but not those without the disease, is allele 2. That is, allele 2 is associated with early Alzheimer's. Unfortunately, the male indicated by Z has this allele. Question 8. Is the SNP1 genotype shown for an individual? GG, GA, or AA? Assume C and G have been labeled with green fluorescence and A and T with red fluorescence. As the spot shows both green and red fluorescence, this individual is heterozygote at SNP1, that is, the individual has genotype GA. So that's genotyping in brief. Give the video a thumbs up if you liked it, and don't forget to subscribe to be informed of new videos on related topics. Thanks for watching.